to be working. All right. All right, before we get started officially, are there any announcements from faculty or anybody else? ...2022 seminar series. And today's kind of different because we'll be hearing from one of our own, actually 4 of our own. The first 2 speakers will be talking about their research projects that they've conducted over the past year for OS 400-401, the capstone project that each and every one of you goes through. And then the last 2 speakers will be talking about internships that they were involved with during the summertime, last summer. So at this time, I'm going to introduce the first speaker, which is Gloria Passman, and the title of her talk is Investigating the Antimicrobial Activity of the Marine Sponge Haliclona oculata.
Good afternoon, everyone. Can you hear me all right? Yes. All right, as Professor Verdi introduced, I had spent my senior research looking at the marine sponge Haliclona oculata, which is a common marine sponge found locally in Castine and greater New England. So in my study, I had aimed to see if the sponge contained compounds that could be potentially used as an antibiotic. This is something that is being currently studied globally in the Pacific and mostly tropical waters, partially due to ease of access. A lot of these species, when they were extracted and used in an exposure, such as you can see on the left side here, against various model species of bacteria, such as Staphylococcus aureus. So for this particular study that I used as an example, they had looked at marine diatoms and performed an extraction process in order to determine whether these had expressed an inhibitory reaction to the bacteria. Where you would see that would mean, that the bacteria were either killed or they were unable to grow due to the presence of that extract. In the case of this study by Kin et al., 2013, they had scooped out wells in an agar plate and exposed the bacteria to various diatoms that they had excluded and made sure that they had pure culture before they conducted their test, which is something similar to what I had done. In the end result, they had taken all of their data and had found the measured zones of inhibition to have a quantitative way to determine whether one was more effective than another and to see, if one of them had the greater potential for reuse as these antibiotics. Recently, there has been a rise in the antibiotic-resistant strains, which makes this incredibly important in terms of the research world in order to develop these new antibiotics to help cure illnesses that are often caused by bacteria. So, in order to do this with my own, I had to extract the sponge with the help of the scientific divers on campus to gather my samples and begin to extract them. Of course, first off in the process for me was making sure that they were the correct samples. So I had to use bleach, which you can see on the left, in order to make sure that the sponge was the correct species and was all the same, because they have a very plastic way of growing where one can look a lot like the other. On the right is a clipping from one of my samples, and you can see they're pretty, nothing really stands out characteristically when you immediately look at it, which is why it's very important to double check this. When it came to actually extracting my samples, first I had to take the samples, identify them, and then prepare them for preservation until I was able to go through the extraction process. In doing this, I would flash freeze them to -80 Celsius to prevent any potential continuation in any processes that the sponge would undergo, be it growth or stress hormone release, that could potentially influence the results. From there, I would go into the extraction process of exposing the sponge to methanol and using a vortex to work on breaking up these chunks of sponge into a extracted slurry, which I could then begin to dilute. And this dilution process is important to be able to, should I have had an inhibitory response, see if that increased with concentration or there was potentially other factors to be included. In doing this, I had diluted it into a low, medium, and high concentration, which would give me a result where you would ideally have one result that had only a small ring where you didn't have any growth, a medium getting slightly larger, and then ideally the high concentration, which would have the biggest inhibitory ring. For my own study, I had exposed Helicona oculata to Escherichia coli, which served as a example model species for gram-positive bacteria, and then Staphylococcus epidermidis as my gram-negative, which are both two classifications of bacteria which were defined by their cellular membrane, in that the gram-positive would have a thicker membrane that would potentially influence whether these inhibitory compounds would be able to influence this bacteria. Once I actually had completed my study using the Petri dishes similar to on the prior slide, we found that there was an increase in inhibitory response depending on the concentration. Using a photospectrometer, I was able to calculate the exact extraction amount of light absorbance for each of my low, medium, and high concentrations, so I could better qualify whether the inhibitory ring had a correlation with the increasing concentration, similar to Kin et al.'s study. So in this particular figure, you have the line going increasing over with the increasing concentration, similar to that prior figure. After the conclusion, with some statistical analyses of a ANOVA, we were able to find that there was a significant difference between the high concentration of my sponge extracts and their inhibitory response viewed versus the medium low concentrations and my blank, which served as my standard to make sure that my paper discs that I put my extracts on did not influence my results. An example that I had used in comparison for this was with gentamicin and vancomycin for each of my bacteria. I did not see any significant results with my Staphylococcus aureus, or epidermidis, excuse me. So I wasn't able to do this comparison because there was no response. Whereas in E. coli, there was a significant response that could be observed where that high concentration was able to have an inhibitory response that, at least, looking at the mean inhibitory ring diameters, was relatively similar to that of the gentamicin as the known antibiotic. However, with greater variability compared to the gentamicin. In the course of doing these, not everything was entirely smooth. Not every result was a smooth line that you could see. And because of that, there is a greater variation between my samples and their consistencies. Over the course of my study, I had also allowed my Petri dishes to, in the incubator, sit for 24 and 48 hours. And in order to see whether that time difference had made a significant change in the response that was seen. However, due to varying difficulties, be it with the cell densities and the potential ability for the bacteria to have been smeared due to remaining moisture and things like that, there was no change visible there that I could potentially look at. So overall, the E. coli had shown the greatest response to my sponge and gives it to be an example of a species that could potentially be looked at in the future in order to develop these antibiotics that are desperately needed in the medical field. I just wanted to give a quick thank to Pam Grindle, who was a great help in getting the lab set up and prepared, Professor Alan Verdi and Carrie Friedman for their help in the process of going through senior research and getting everything set up, because that is a very big project, as many of you will find out very soon. And also Professor Whitney, who was my project advisor, and of course Campus Safety and Amber Durant for their help in getting into the lab at very odd hours to make everything possible. So.
All right, we have a couple minutes before we turn over to the next speaker. So does anybody have any questions?
Hi. So other than this being like a topic that's like really needed soon to help with medication, is there any other reason why you like picked this topic specifically?
Well, a lot of it came into availability of microorganisms and also their study in, our local area, because there's been a lot of research into things such as marine alga. So we've had studies in the past by students such as Carly Karen in 2010, who had a similar project to mine where she was looking for antibiotic properties that could be found in Fucus fasciculosis and similar macroalgae. And so there is levels of inspiration from that as well, but I wanted to look more at, species that aren't necessarily immediately prevalent, but have also shown great promise as the marine sponges have in the tropics where there has been more research. Does that sort of address that question, or is that?
Any other questions? We have time for a quick one. All right, at this time we're going to change speakers and turn it over to Lauren Plissy, who is going to be talking about evaluating the relationship between salinity and total phenolic concentration of Fucus fasciculosis along the Penobscot River, Maine.
Yeah, hello. So my topic, like Professor Verdi just said, was with Fucus fasciculosis, phenolic concentration, and the impact of salinity on that. Fucus fasciculosis is a common macroalgae found along the intertidal zone of Maine's rocky coastline. The intertidal zone is an extreme environment with drastically changing parameters such as temperature, wave action, light exposure, and salinity. And many organisms in this environment depend on macroalgae to survive, but macroalgae produce their own chemical that aids in defense against herbivores in these changing parameters known as polyphenols, which have antioxidant capacities. And brown algae produce a common and specific type of polyphenol known as fluorotannins, which is this compound, and that can make up to 20% of the algae's dry weight. And studies by Berkemeyer et al. And Menkenek et al. Have quantified and identified phenolics within the tissues of algae. And then other studies by Lomatier et al. And Van Ees et al. Have looked at the environmental and biotic factors that impact phenolics within algae. But understanding the variability of these chemicals, especially in a dominant alga such as Fucus fasciculosis in the Gulf of Maine, that has a changing climate like increased frequency and intensity in storm events, is important in understanding their either impact and ecological structure. The primary goals of my study were to determine if there was a significant difference in mean total phenolic concentration of Fucus vesiculosus, and I also determined if there was a significant relationship between salinity and total phenolic concentration. My methods were derived from this Van Hees paper, and I made a few modifications, mostly just to the volumes of my extracts and samples. First, I collected 15 samples randomly along these sites along the Penobscot River. There's 5 of them varying in salinity, and I just chose them based on their variation in salinity, and then also accessibility and permission to collect. And my lowest saline site is Bucksport at 18 parts per thousand, and my highest was Fort Madison and Castine at 34 parts per thousand. So once I collected my samples, I froze them at minus 80 degrees Celsius until they could be analyzed, and then when I started to analyze them, I used their apical meristem tissue, ground it under liquid nitrogen, and then extracted it in 80% methanol in the dark for 24 hours. After the extraction, I created a fluoroglucanol standard. Fluoroglucanol is a monomer of polyphenols, so I created that standard with known concentrations of fluoroglucanol and created a standard curve that I later compared my samples to. I added a Folencio Cal2 reagent, which binds to the polyphenols through a reduction reaction, which is this in the top left-hand corner. That was added to my extracts and my standards. And then after that, I added sodium carbonate to create an alkaline solution. I diluted my extracts with type 1 reagent grade water and heated it in a water bath for 30 minutes at 50 degrees Celsius, which is this picture on the right. And I read my standards and extracts at 765 nanometers on a spectrophotometer to gather their absorbance. And finally, I calculated the phenolic concentration of my samples using their absorbance and the linear regression equation from my standard curve. So my study revealed that total phenolic concentration of Fucus vesiculosus was highest at Morris Cove and Bucksport, and when compared to all my other sites, and it was highest at Morris Cove with 11.37% fluoroglucanol per wet weight, and lowest in Castine at Fort Madison with 2.85% fluoroglucanol. And then I also found that there was no significant relationship between salinity and total phenolic concentration of Fucus vesiculosus. In contrast, one of the highest concentrations was at my lowest salinity site. And one of the similarities between my 2 highest phenolic concentration sites, Morris Cove and Bucksport, was anthropogenic activity. This includes vessel operations at Morris Cove and the paper mill in Bucksport. These emit aquatic pollutants, which in turn impact the nutrient availability and heavy metal concentrations for intertidal algae. And copper is a metal that is a micronutrient for macroalgae, but at high levels can be toxic. And Conan and Stengel found that increased copper levels induced oxidative stress in algae, and then in response, algae produced antioxidant capacity compounds known as polyphenols in response to that. But there's more research that has to be done into the metals in the water and in the sediment and the tissue of the algae and the relationship that that has with phenolic concentration, and there could be a possible relationship between those. And then for my second objective, that was in contrast with papers I'd read and based my hypothesis on. I found that there is no significant relationship between phenolic concentration and salinity, but my paper, Van Hees and all, where I also based my methods off it, found a positive relationship between the two in 2 brown algae in Western Australia. And so one reason for this difference could be species-specific, like intertidal algae's tolerance to salinity. A paper by Back et al. Tested the salinity of cultured Atlantic Fucus vesiculosus. They tested low salinities from 1 to 5 parts per thousand. to a high salinity of 45 parts per thousand and found higher growth and higher survival at higher salinities. And the estuary that I sampled had a salinity gradient that ranged from 18 to 34 parts per thousand. So that falls right in the range of bacchanal. And like I said earlier, these phenolics are antioxidant compounds, and with higher salinities could induce oxidative stress. But based on what I found, Fucus fasciculosis tolerance to salinity did not induce oxidative stress at these locations. And this study just provides baseline on phenolic concentration of Fucus fasciculosis along the Penobscot River in Maine. It's really, the data has been lacking for this, and I think this is an important research just in providing baseline data for impact evaluation, especially in a changing climate and important systems like estuaries. And I would just like to thank Professor Mullen, my project advisor, and Pam, our lab manager, and then also Professors Verdi and Friedman and Professor Baer, and my OS 401 colleagues. And thank you.
Questions that you would like to ask Lauren? Sarah?
Given if you could redo the experiment or like do further studies, what if you wanted to try to control for like anthropogenic activity?
I think so. I think this was really good to know because other studies had seen a correlation between salinity and phenolic concentration, but it definitely would be cool for like someone else to do anthropogenic like metals and stuff like that. But I would have done it too, probably, if I knew. But yeah.
Is this on? Yeah. So you answered this, but one thing that will engage the algae into developing these phenolics is predation.
This past summer. So Phoebe is going to be talking to us about seizing opportunities from the Atlantic to the Pacific.
All right, can everybody hear me? All right, awesome. So I actually ended up taking a bit of a different approach to my presentation and decided to talk about all of the both formal and informal internship opportunities I've had so far at MMA. So I'm going to start off by talking about summer 2020, where I had an internship at BOOTHBAY Science Center as a science instructor. And so basically what we did that summer was that it was it had an educational focus, and the program ended up going online. And because the program went online, we ended up making videos throughout the summer on activities and such. So sorry. Nope. Wow, I'm struggling. That's okay. Anyways, so we ended up having to do the program online, and because of this, we still ran our touch tank, and we did a bunch of educational videos on the touch tank. So you can see me over there, and we used locally collected organisms from the Gulf of Maine. In addition to just working on the education side of things, I learned how to sail during the summer on the turnabouts, and we ended up making a rack, which you can see in the middle photo, as well as learned how to operate whalers in the marina and up and down the Damariscotta River. And aside from doing the educational piece and then the boating piece, we also did a lot of maintenance that summer. So I thought it was just really interesting to include that into the study, or into the presentation. And then more excitingly, this past summer, I went with Professor Verdi to Walla Walla University's Rosario Beach Marine Laboratory in Anacortes, Washington, where I worked as both a lead aquarist, a scientific diver in training, and a water quality analyst. So as the lead aquarist, it was actually originally a self-employed title, but I did live up to it, and I was responsible for taking care of up to 30 individual octopus at a time, and as well as some other invertebrates that we used for feeding, such as purple shore crabs, as well as clams. And so basically what I did as the lead aquarist is I would do daily feedings as well as daily health assessments of each octo, making sure that they were okay, that their behavior was normal, and that they had no signs of senescence, which is essentially tissue deterioration due to age. And I also ended up, blinking. What I also did as the lead aquarist is that I was constantly maintaining the seawater system and making sure that the valves weren't blocked and that the pipes weren't blocked and that the seawater was flowing, because we did have some issues with that. But, oh, there we go, there's the laser, sorry. So the first octopus I wanted to talk about is the moose octopus, and I was actually the first person to get them to consistently eat in the lab, and they were being studied for their burrowing activity, which is super unique to cephalopods. They are one of the 2 known species to actually burrow in the mud, so that's what they were being used to study for this summer. The giant Pacific octopus, this is actually a juvenile, and it was accidentally collected during one of the grad students' dives, but we ended up having a few in the lab. And then lastly, the octopus rubescens, also known as the ruby octopus, was we collected for denning activity, as well as more specifically the project I worked on was for ocean acidification, which I'll get to in a minute. But yeah, so basically the biggest part of my summer was that I was an AUS scientific diver in training, so that is the American Academy of Underwater Sciences. And so I decided to put a bunch of visuals up. So this is actually a picture from Professor Verdi of a Puget Sound king crab juvenile. And the other ones are just common things that, well, actually uncommon critters that I would see while I was diving. But as a scientific diver, I did 71 dives this summer, 46 of which were scientific specifically. And so those included transect dives for density bottle preference data for O. rubescens, which was this guy, as well as collection dives of O. rubescens, which we would bring back to the lab or the grad student would use. Professor Verdi and I also went on camera deployment dives where we would bring equipment down for a grad student. And the moose octopus, right here, is what we used. We collected them at night, so it was actually a bunch of night dives that we went on, and we'd go out and we'd take our little flashlights and we'd hunt for them and try and grab them on the sediment. It was really interesting. We also ended up going dumpster diving for bottles, and we would place those at different locations to promote denning. And lastly, one of the most memorable things we did was we ended up cutting PVC pipes and made little houses to promote selective size denning of O. rubescens. So who knows, the students this summer might find out if that was effective or not. But yeah, so scientific diving was a huge part of my summer. And then the most formal setting that I had this summer was that I worked in the lab on an ocean acidification project by a visiting professor at the laboratory. And basically what I did was I was taking daily seawater samples from the OA room. So the OA room was in the wet lab, and half the tanks had the control, and then half the tanks were experimental. So the control tanks were bubbled with CO2 for the correct concentrations if you were just go and take seawater out of the ocean where the octopus were found, or they were bubbled with a higher concentration of CO2, which therefore would increase the acidity of the water. And basically what I did was every day I'd go down, take my water samples, bring them back up to the lab, use the spectrophotometer, get the spec pH, I'd toss the numbers into R, and then they'd spit them out in an Excel sheet, and that basically told us what the pH of every single tank in the lab was. And then also every week I had the pleasure of doing titrations, which was so wonderful, absolutely. It was a hoot. I dropped a lot of glassware this summer too, so I certainly am not big on lab stuff, but I ended up learning a lot. And basically what we were doing with the titrations was that we were trying to find the alkalinity of the seawater. And once I got all the seawater samples, bring them up, titrate them, and then I got to input all the data into these awesome little boxes that Dr. Onthank actually created, who was the director of our RBML this summer. And so basically the whole purpose of this study was so that in the end we could draw blood from octopus and basically see how it was affected. I wasn't really part of that. I mostly just did the lab stuff, but I did successfully once draw blood from an octopus, and it was actually quite difficult. But yeah, so I had a great experience this past summer, and that's been the most like science-y internship I've had. And then the last thing I wanted to talk about is that this winter I actually had a more informal internship at a local aquarium store. So over Thanksgiving break, I noticed that they had come in, and I decided to ask if I could intern there, and I ended up learning a lot. And so this is a piece of bird's nest coral that I learned how to frag. I learned how to take care of and transport corals and make sure that they were slushed away with the diseases before they were put into our main tanks. But yeah, so basically just wanted to say that internships don't necessarily have to be in a lab. Like, it's basically make the most out of the experience that you're presented with and try and enjoy and what you like to do. Yeah.
Questions?
Hi, Phoebe.
Hi,
Love
Courtney.
the presentation. I was just wondering, where did you hear about all these internships? Like, I know BOOTHBAY Sea and Science comes to our career fair, but I haven't heard of any of these other ones.
Yeah, so BoothBay Sea and Science Center, as Courtney said, they come to our career fair. Professor Verdi actually reached out to me and asked if I would go to Washington with him. And then I actually went out of my way and kind of just asked the store owner at Corals Unlimited if he would be interested in having an intern. So basically, like, you can go onto REU websites through the National Science Foundation, but some professors take students every summer, or you can just kind of find them on their own. Aquaculture is a big one right now too that students are doing internships at.
A lot of big aquariums have difficulty managing just one octopus, even with their budget. How exactly does one keep a hold of, you said 30 octopuses?
Yeah.
How do you do that, since they're known for being escape artists?
Yeah, so I couldn't really hear what Ben said totally, but yeah, so 30, basically they were called octopus condos, and I can go back. So they were small containers. So the moose octopus were actually they're like itty bitty, and then O. rubescens are also relatively small, but they were constantly being released, and then new octopus would be brought into the lab. But yeah, so they were in smaller holding containers in the wet lab, so that they wouldn't escape. I don't know if that answered your question.
Hi, Phoebe.
Hi, Callie.
So I don't know exactly where you're from, but going to Washington for an internship, how would you just describe that experience? Because most of our internships are on the water and aren't very close, or could be close, but there are a lot of ones that are California, Washington, all of that, the West Coast. So how did you feel about that?
Yeah, so I'm from the Mid-Coast Maine area, And so I was actually extremely nervous to go across the country because I hadn't really been away from my family that much before. And I thought it was a really great experience, and it was nice to be like somewhere new and to have such a great mentor to like show you around. So just make the most out of the opportunities, even if it's like scary to apply somewhere far away.
Hi, Phoebe.
Hey.
So I had a question. How would you go about, like, are there different processes for applying for like a formal internship versus informal, and like how do you go about doing that?
Yeah, so BOOTHBAY and Science Center, I'll just scoot back, that was, they came to the career fair, and I talked to Pauline and set up an interview and such, and ended up landing the internship for the summer. So a lot of them, a lot of places are like online applications nowadays, and you'll send it out, and usually they'll send out for interviews. But for like my informal one, I straight up walked in the store and was like, hi. I was like, would you be interested in having an intern, totally unpaid? I just really would like to know more about the aquarist hobby and how corals work. And so they were super willing. So I think if you have enough confidence just to give it a shot.
All right, thank you very much, Fee. Our fourth and last speaker is Ellie Jellerson, and she's going to be talking to us about her internship at Texas A&M REU, observing the ocean.
So this past summer, I did an internship through Texas A&M, which was called Research Experience for Undergraduates, Observing the Ocean, and it was supposed to be in person. So typically you would travel to these locations and they provide you with housing. And this one was supposed to include a research cruise, but because of COVID, it got moved online. So this is a photo of my cohort and all of our research advisors, and this was a lot of my summer was Zoom meetings with all of them. But REUs are a great internship experience. They're typically 10 weeks, and they're research intensive. But because this was virtual, a lot of our projects focused on data analysis from previous projects and coding. It's also paid, which I know is a big thing for people for summer jobs. So all of these REUs are paid pretty well, and I also received a technology stipend where we were at home. And then they kind of accumulate in a final presentation of what your research project was for the summer and a research paper. And so this screenshot is of what my computer screen looked like for most of the summer. I mostly worked in R to create a machine learning framework, and I entered the summer with absolutely no coding experience and was really intimidated and kind of overwhelmed. And then I ended the summer by coding a whole website. So you definitely can learn a lot, and I know a lot of you are in DAS right now with R, but there's a learning curve and you guys will get it. So my project was titled Using a Data-Driven Approach to Predict Chlorophyll Content in the Northern Gulf of Mexico Through River Nutrient Data, and my advisor was Dr. Xuan Zheng. So coastal waters are really susceptible to eutrophication because of their proximity to anthropogenic activities. And this is a really pertinent issue for Texas A&M because of their proximity to the Gulf of Mexico. So the Mississippi River has the largest drainage basin in the United States, and this is where most of our food is grown. So there's a lot of fertilizers put on this land that are rich in nitrogen and phosphorus. And this nitrogen and phosphorus washes into the Mississippi River and is then carried to the Gulf of Mexico and kind of dumped there. And when it is dumped there, that causes intense phytoplankton blooms, which ultimately use up a lot of the oxygen in that area and create the Gulf of Mexico dead zone. So the basis of our project was we wanted to be able to predict chlorophyll content from this river nutrient data in the hopes of maybe providing some information on how big these blooms could be, and then maybe knowing when these dead zones were going to occur. So to do this, we built a machine learning framework, but machine learning is a really abstract concept to most people, so I just want to walk you through like a brief overview of what you have to do to make those networks. So first, you have to get your data, and we were working with tens of hundreds of gigabytes of data, so the more data, the better, because there's a lot of power in numbers. So we collected data from all over the Gulf of Mexico, And then you have to clean, prepare, and manipulate that data. And so in order to do that, you have to perform some temporal and spatial bins and make sure all the formats are the same and make sure you have all the data in the same sheets and ways that they can be analyzed together. And so these first two steps were what a majority of my summer was. So I would say the first 7 weeks was mostly on obtaining that data and then cleaning, preparing, and manipulating it. But then once you have all that data in that really nice clean form, you can train your computer model. So you give a computer 75% of your data, and it generates an algorithm, and it looks at all the relationships between your parameters and tries to make a prediction algorithm. And once it has that algorithm, you can test it. So you use that other 25% of your data that you didn't give the machine to make an algorithm as a test. So you know what actually happened from this data that you starved the model from, and you compare what actually happened to what the algorithm predicted happened. And then you can always go back and improve upon these things. So I collected data from the United States Geological Survey stations and the Gulf Coast Ocean Observing System to fuel the machine learning framework. And so I collected data from 4 different rivers, and they were the Mississippi, the Atchafalaya, the Brazos, and the Sabine, and they're the 4 largest rivers that drain into the Gulf of Mexico. And then I collected all these little points, which are for chlorophyll data. And so these are under spatial bins, but I also collected data on temperature, salinity, oxygen content, pH, and chlorophyll. So I made this kind of plot for all of those different parameters. But then I wanted to see how river nutrients impacted chlorophyll. So I also had to collect river data from those 4 rivers. And so I collected nitrogen and phosphorus content, and you can't really see the difference in colors on these graphs, but the dots indicate actual observed values, and then I used a computer software to extrapolate between those points so we could get a continuous daily value, so we could line it up with whenever those chlorophyll or temperature or salinity measurements were also taken. What we wanted to do was to pick specific ocean stations that had really complete data sets and be able to tie them to specific river stations. And so the figure on the right, or I guess your left, shows all the individual data points for chlorophyll that we had. And that's a really messy figure, and it's hard to look at, and it's hard to pick out a specific station. So we binned them into 0.2 degree latitude and longitude cells using R, and then we get a much prettier figure like you see on the right here. And then we can also easily see which of these stations, based off of their color, had the most amount of data. So we did this type of binning and plotting for all of the individual parameters we had. And after all that work and 7 weeks of collecting over 500 gigabytes of data, we only had 2 stations that had enough data to fuel a machine learning framework. So those were stations 1587, which was close to the mouth of the Mississippi River, and station 3061, which is off of the west coast of Florida. And so we use these 2 stations to build our machine learning framework. And we tied them specifically to the Belle Chase, Louisiana River Station, which is the Mississippi. So our model training was done using the random forest algorithm, which there's many different algorithms that you can train machine learning frameworks with, but that's the random forest algorithm is the one that we chose to work with. And so for station 1587, our testing data was able to generate an algorithm that could predict 94% of the variation in our data, and in 3061, it was 91%. But then we wanted to see if river nutrients were the best predictor of that. So we looked at the order of variable importance, which is looking at which variables are the best predictors for chlorophyll content. And we found that for both of the 3 stations, the river nutrients and the water temperature were the most important, which makes sense because these fluxes of nutrients are typically seasonal. So that would correlate with the water temperature. But the slight difference are likely because of the differing distances from the river mouth, and not including river nutrient data from rivers near Florida, which are likely having local effects. But ultimately, there's really complex interactions between nutrient utilization and availability in these areas, and a lot more data and study is needed to understand the full extent of these interactions. But if you think the project was interesting, I'm also presenting this at ASLO this March virtually, so that'll be online. But I also built an interactive website as a final part of my project. So it has all of this data, and you can click through it and look at different time series if you're interested, and the link is at the bottom of this slideshow. Thank you very much.
All right. We have a few minutes here for questions before we have to turn the room over to the next group.
Do you think if you would have been in person and like collecting the samples there, your data would have changed? Or how did you, like, the data that you were receiving, when was it collected, and was it, how do you know that it was like the most accurate data that you could have received?
Yeah, that's a good question. So the Gulf Coast Ocean Observing System is this platform where everyone who does research in the Gulf of Mexico that is published in studies has to submit their raw data. So you can go there as anyone here if you wanted to do a project on the Gulf Coast and download all this data. And I was working with data from as early as 2000 up to as recent as a month or two before I started my project. So it's a really wide range, but it was a really reliable source because all of this was from peer-reviewed studies.
Anybody else have a question? Sarah.
Because you'd get to get experience doing this sort of stuff, or is it like, oh dear God, what have I done type thing?
It was definitely an oh dear God, what have I done type of thing. So I applied right before COVID, and I was supposed to go the summer of 2020 and do a totally different research project, which I actually based my senior research project on. And then they paired me with Dr. Zhuang, and he said it would be really R-heavy and coding, but he said it was really valuable. And so I kind of just took the leap, and he said you would figure it out, and it was, even though it looked complicated, it wasn't that hard. So there is a learning curve, but I don't know if I would have applied specifically for a coding internship because I wanted the lab time, but I'm still really glad I did it.
One more question? Anybody? All right, well, thank you, Ellie. And this concludes our seminar for today. So we'll see you whenever we have our next external speaker. Thank you.